Hey guys, in this video we're going to recap over the different structures that DNA can make and the process of the cell cycle and mitosis. So first we have to go over the word chromatin. If you look at this picture here, in this nucleus, you might see that there's not much going on in there, it appears. And if you look over here at this picture, you can see that this is, in these strands, it appears that there's these little beads in there. And what this is this is chromatin. Chromatin is referring to two molecules. It's referring to both the DNA strands, okay? And it's also referring to the proteins that the DNA is wrapping around. Those proteins are histone proteins. And in chromatin, the DNA is loosely wrapped around the histone proteins. So in this nucleus that you see over here, it doesn't look like there's much in there. That's because that chromatin is not tightly wound up. It's not able to obtain any stain if a scientist were to try to stain it. If we go to chromosomes, chromosomes are tightly wound DNA around the histone proteins. So we can see this is the strand of DNA, and it starts to coil around the histone proteins. And then those coils of histone proteins continue to coil around each other until you get this structure of a chromosome. And now if a scientist were to stain the nucleus of a cell with chromosomes with tightly wound up DNA, since those are so condensed, that would hold on to the stain, and the scientist would be able to view those under a microscope much better. This would be, I believe, using an electron microscope to view the chromosomes. And essentially all a chromosome is condensed and tightly coiled DNA. And in a little bit we're going to go over why the DNA has to be tightly coiled up and condensed for a specific purpose. One thing to consider is these two structures here both represent one chromosome. Now this one right here actually has a duplicated copy. So this has two identical copies, but since they're attached together, we only consider that one chromosome, even though that is essentially two copies stuck together. Okay. Next, we have to go over what a sister chromatid is. The sister chromatid is each identical half. So again, this whole structure is one chromosome, but if I'm referring to the left or right half, that would be an identical sister chromatid, okay? And next, we have to go over the centromere. Essentially, the centromere is just where the two sister chromatids attach. That's the center point, okay? Next, if you look at this photo, which we went over in class before, what this is a tissue from an onion root. And this onion root is, the cells are going through different stages of mitosis. And if you look at the different images, like this one right here, you could see that the chromosomes are tightly condensed. The DNA is in a chromosome state. And if you look here at this image, the DNA is not very visible, and that's because that DNA is in a chromatin state. It's not very condensed, so it's not going to retain those stains that the scientist is putting on the slides. So why do scientists understand cell division? Well, if you remember from class, we talked about three important things. Number one was for growth of new cells, for growth and development. So at the beginning of everyone's existence, we all started off as one single-celled zygote. And over time, we've developed into adults or young adults, which consist over 10 trillion cells. So essentially, it wasn't one cell getting larger and larger. It was the cells growing and splitting, growing and splitting, growing and splitting until we get more cells. So the growth of brand new cells for growth is one reason. The second reason is replacing old or damaged cells. If you get a boo-boo, those cells are damaged. You got to replace those. And over time, with just aging, cells, they get worn out. And then after they have done their job for so long, the body has to get rid of those cells and replace those with new cells. Both of these processes, the cells get a signal to stop growing and stop dividing at some point in time. And if a cell doesn't get that message to stop growing and dividing, then that's what's called cancer. Cancer is called uncontrolled cell growth, okay? They just don't get the memo to stop. Different cells divide at different rates. So if I'm looking at a skin cell, these cells that are deeper, these cells at the deeper level, come on now, over here, these cells that are deeper, they go through mitosis at a much higher rate than most other cells, okay? So your skin is constantly sloughing off on top, and since those cells are sloughing off, they constantly have to be replaced. So the cells in the bottom are going through mitosis at a very fast rate to replace the ones that are sloughing off. If we look at intestinal cells, those cells go through mitosis once every 3 to 5 days. Those cells have to be replaced because there's a lot of stuff going on in there. There's a lot of sloughing around, a lot of cells getting etched off, and so small intestines, they go through mitosis at a much higher rate. If we look at nerve cells almost never go through mitosis, and that's why nerve damage usually doesn't heal itself because those cells don't grow and replace each other. If we look at this chart right here, this chart shows the typical cycle of a cell. If you notice, the longest part of the cell cycle is interphase, and the DNA during that whole time frame, the DNA is in chromatin. So most of the time, if you look through a microscope and stain it, you're not going to see much DNA in there going on or see any chromosomes. After interphase, the cell goes through mitosis, and mitosis is the division of the nucleus. So we're trying to divide the DNA equally to equal parts. And then finally, cytokinesis. Cytokinesis is division of the cytoplasm, all the cell stuff, all the cell juice, everything else. So if we look closer at interphase is divided up into 3 parts. We have G1, and G1, if you read it in a textbook, that stands for gap 1, but I like to think of it as growth 1 because the cell is going to grow. So in G1, the cell grows. There's a high amount of protein production, okay? Next, we go to S phase, and S stands for synthesis. In synthesis, DNA is going to be replicated. So we're going to get another copy of every chromosome. Each of those copies, they're going to end up attached to each other at a centromere. And I'm going to show you guys a picture of that in a little bit. Next, we go to G2, which is a second growth phase. And what's unique about this is we get an extra centrosome. And the centrosome is what's going to be doing the actual division of the chromosomes. After interphase, we go to mitosis, which is division of the nucleus, and all of those processes are PMAT. And then finally, we go to cytokinesis, which is division of the cytoplasm. So the cell cycle, here's what it looks like. We start off with interphase. We're going to have our cell. It's going to grow a little bit. We have a lot of protein production going on. Next, we move on to S phase. If you noticed, during that process, I doubled up on the DNA in the nucleus. Okay, so there's the DNA replication. Next, we go to G2, and in G2, we got another centrosome. Okay, we replicated that centrosome. Following interphase, we start mitosis. The first process, or the first step of mitosis, is prophase. Now watch what happens in prophase. 3 things are going to happen. We have the sister chromatids. First, the DNA, the chromatin, is going to condense into chromosomes. Each chromatid is going to pair up with their identical match to form the chromosomes right here. Okay. The centrosomes move the opposite ends, or they start moving to opposite ends, and the nuclear membrane breaks down. Okay. Following prophase, we go to metaphase, and metaphase stands for meet in the middle. Once all the chromosomes are lined up in the middle, you have these spindle fibers, which are made of proteins, that they're going to come out and attach to each sister chromatid. Okay. After metaphase, we go to anaphase. Those spindle fibers start to pull the sister chromatids apart, and also the cell starts to elongate. It starts to look more like a football shape. Okay. After anaphase, we go to telophase. And remember, anaphase, real quick, anaphase, the A stands for apart or away. Telophase stands for 2 nuclei. So we're going to get the 2 nuclei start to reform. Those chromosomes are going to decondense. They're going to go back to their chromatin state. Okay. And essentially, if you want to think about telophase in an easy way, it's prophase backwards. Okay. Following telophase, well, actually, telophase and cytokinesis, they kind of happen at the same time. but they're two separate processes. In cytokinesis, we have division of the cytoplasm. And that's going to happen, let me backtrack just 1 second. That's going to happen by the membrane forming what's called a cleavage furrow. So this membrane is going to start to pinch in, and once it splits down in the middle, we get our two cells that break apart. And we're left with two identical cells. Okay, so one key point in mitosis is we start off with a cell that has 46 chromosomes. Now, what are we going to end up with when mitosis is finished? We're going to end up with two cells that also have 46 chromosomes. Okay? So each daughter cell has the same genetic information, has the same copies of DNA. Now, you might be asking, if this cell cuts in half, how did these two cells end up with 46 chromosomes? If this original one splits in half? Well, here we go. So we have our original cell with 46 individual chromosomes. Now remember what happens in S phase. During S phase, DNA is replicated, and those identical copies are going to be attached to the chromosome. Now, what do we call, even though that's two copies of DNA, what do we call that? That's still considered one chromosome. So that's still classified as 46 chromosomes. Now, when these two halves split apart, each cell has one copy. Okay? In short, the cell cycle, the entire life of the cell, starts off with interphase. We go to prophase, metaphase, anaphase, telophase, cytokinesis, PMAT, if you want to remember just the steps of mitosis, PMAT, division of the nucleus, and the end. That is mitosis, and I hope that was helpful.